Hey everybody, welcome to the fourth Connectome podcast. This time we've got a real treat for you guys. We're joined by Scott Emmons, who's a professor of neuroscience and of genetics at Albert Einstein College of Medicine. And Dr. Emmons is on to talk about a really interesting paper he's just published following up on some Nobel Prize-winning research on the nervous system of a roundworm known as C. elegans. You might have heard me mention that in some other articles. Now, the C. elegans connectome has actually been mapped since 1989, but we're only recently beginning to understand what exactly a lot of the networks in its nervous system do in relation to one another. Dr. Emmons' new paper focuses on several different kinds of feedback and feed-forward loops in the C. elegans connectome, and he's actually got some very detailed, data-driven theories about what types of loops help a nervous system lock in on certain kinds of behavior. And I'll let him tell you more about the details of what that means.
So to start, let me just ask you to talk a little bit about yourself and how you came into connectomics research.
Okay, I have a background in basic genetics and molecular genetics and molecular biology going back to the early days of recombinant DNA research. Those methods were being developed, and I studied at that time phase lambda, and we're interested in how genes determine the phenotype of the organism, whether it's a virus or a bacterial cell or a higher animal. And so then on my Ph.D., I went into developmental biology with an interest of trying to understand how the genes determine the phenotype of the organism. And to tackle that problem using genetics, I decided to work on the nematode worm, C. elegans, which probably you know has many research advantages for doing genetics research. And it was an exciting new field, small field at that time in the late 70s, and I've been working in that field ever since then. In the mid-80s, I turned my attention specifically to studying the male C. elegans as a focus for developmental studies. And so for many years, we actually mutants that affected development of the male, and studied cell fate specification and sex specification and aspects such as that. And as we were doing those experiments, more and more, we had mutations that affected the male's behavior, particularly sexual behavior. And so a little over 10 years ago, I realized that an important missing piece of information about C. elegans was the wiring diagram of the male, and it was something that we really needed to go further with studying the genes that affected development of the nervous system and behavior of the male. So I began thinking about how to tackle that problem of getting the wiring diagram of the male. I knew the individuals who had done the hermaphrodite wiring diagram at that same MRC laboratory in Cambridge, and they advised me and talked about their prior efforts to do the male, which they were unable to complete because they just found that the neurons were branching and there were extra neurons in the male, and the male nervous system that governed male cognitive behavior was too complex for them to reconstruct in the same way that they had reconstructed the hermaphrodite nervous system. Somewhat simpler properties of mono-directed neurons, single process neurons that run in stable bundles that don't move around too much as they navigate through the body, made the reconstruction possible. The male was more like a normal nervous system with neurons going every which way. So it turned out that my colleague here at Einstein, David Hall, is an electron microscopist and has worked on C. elegans and on the nervous system his whole career from the 1970s. And he had taken custody of all the materials from the Cambridge group, starting back in the mid-early 70s. All the electron microscopy they had accumulated, that laboratory had closed by this time, and the only materials had come here to Einstein to be under David Hall's curation. So I had the male nervous system electron micrographs that had been previously made available to me, and Dave Hall's expertise in how to do a reconstruction, and my problem was to figure out how to do something that was too complex to be done by hand. And so I designed software, which we had first by a software company in India, and then developed for a number of years by my own software person. And we were able to do the reconstruction, which we published, as you know, in Science recently.
Mm-hmm. Well, that makes a lot of sense. So that kind of gives the backstory of sort of how you built up to this study. There are a couple aspects of it that I really want to cover that I think are really interesting to the way in which a complex nervous system functions. One of them is small world networks. You mentioned even on the first page of the article that when there's a high clustering coefficient, networks seem to have more local processing going on, and those smaller networks communicate with larger networks, and that's obviously one of the fundamental principles of how connectomics studies connectivity and networks in the brain. Could you talk a little bit about what were some of the characteristics of the small world networks that you guys found in C. elegans in this
it's
study?
really not such a sophisticated concept. You just determine two parameters of the network, the number of steps from one neuron to another. So that means how far distant is one. So if I pick two neurons at random, let's say, or this is just network theory, any two vertices in a network, how many edges, how many steps are there in between the two of them? So that's one number you can come up with. And the second number is the clustering coefficient, how if two neurons are connected to each other, two neurons are connected to a third neuron, the same third neuron, what's the probability that they're connected to each other, making a triangle? So you can run the mathematical formulas and assessments for this on any network. You get these two numbers, and if the clustering coefficient is high and the number of steps is low, and there's a certain criterion that determined, then it's called this so-called small world network. And the small world network concept was kind of a breakthrough in network research. I forget when this was first published in the 70s. This is all fairly new materials, 80s and 90s. The striking observation was that many natural networks had this small world property. A randomly wired up network has short path lengths but doesn't have very high clustering coefficient. It's a very regular network. Can have a very high clustering coefficient, but long paths in between one neuron randomly picked and another. And so a little bit surprising to find that natural networks kind of compromise these two aspects and are wired up in such a way that they can have sort of the cake and eat it too, so to speak. But it's kind of theoretical or hypothetical to say that plus high clustering coefficient means local computation and short path lengths means rapid communication. I mean, it's kind of obvious to say that, but true dynamical demonstrations of this or proof in a natural situation that this is a significant property, it really, to my knowledge, and this is not my area of expertise, hasn't really been explored. So we put these two parameters in our paper because everyone in the field would expect and ask, like, well, are they small world networks, like the phone system and like the power system and so forth? And so we did that, and they are, not unexpectedly, but it's kind of a gee whiz observation.
Sure, we don't necessarily know what exactly each of these small world networks is doing. It's just something that you're going to be looking for in this type of a study. Sure.
That's right.
Okay. Well, another thing that you guys pointed out, this was later in the paper, but I thought I'll just read the quote from the paper because I thought it was really interesting. You said that attractor dynamics are characterized by abrupt transitions between self-reinforcing stable modes of network activity, and I think that's interesting because people often try to compare, the brain to a computer or compare neurons to sort of switches, but what this seems to be implying is that there's something that's not really directly parallel to binary activity going on, but there's more of a system that fluctuates between different kinds of states. Could you talk about that a little?
Correct, that's correct. So are you familiar with this notion of an attractor network? Do you know what that means?
I don't know exactly what it means, no.
So something you have to look up are papers by Hopfield. He's the first person to point out that a complexly cross-connected network of interacting nodes will behave in such a way that sometimes the state of the system, that is, some nodes are on and some are off, let's say, type of network. Some of those states are stable, and and they won't change. They self-reinforce. Because of the feedback and recurrent connectivity, certain configurations, ons and offs, will stick, whereas other configurations will move on to a different one and a different one. Over time they evolve, but the system will evolve, and it will eventually hit so-called attractor, a state that gets stuck and no longer evolves. And Hopfield pointed out that this behavior of a network is kind of like recovering memory. These networks are also called associative memory networks. And it's like when we try and think of something, we try to remember it, and we get part memory, what's the person's name? Oh, it began with M. What was it? Mary, Margaret, Maurice. We recover the full memory from a part. And Hopfield showed that an attractive network will recover the attractive position, like a memory, if it's somewhere in the state space of all these ons and offs that it can wander through somewhere nearby, and then it will automatically evolve and go to the attractor. So associative neural networks, attractive networks, this is a term in artificial neural network theory that came about in the 80s, I think, and caused a great deal of excitement in the field of artificial neural networks because it showed how a network, again, as you say, not digital pathways like a computer, but the emergent property of a whole network can produce things that seem like they might be memories. So it